Hello, everyone, and welcome to today's webinar, Navigating the Opportunities and Challenges in Analytical Development of Flow Cytometry for T-cell Therapy, a primer for cell therapy guidelines and a panel discussion. I'm Marsha Hartman from Slingshot Bio, and I'll be your moderator for today's event. Today's educational webinar web seminar is presented by LabRoots and brought to you by Slingshot Biosciences, Inc. To learn more, visit www.slingshotbio.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box to the left and click Submit. We'll answer as many questions as we have time at the end of the presentation. You may also use the Ask a Question box to let us know if you're having trouble seeing or hearing the presentation. I'd like to welcome our speaker for today's webinar, Therese, Head of Analytical Sciences at Tygen Pharma. For a complete biography of our speaker, please visit the Biography tab on the top of your screen. Therese, you may begin your presentation.
Thank you very much for that. I'm very happy to be here to share. Thank you very much for that. I'm very happy to be here to share my thoughts about navigating the opportunities and the challenges that we have in the analytical development of flow cytometry for T-cell therapy. And I will try and today go over a little bit of background on T-cell therapies and flow cytometry and what we are using it for. What are the opportunities really when we use this for the T-cell therapy in the manufacturing space? From there, I will go into the challenges because we'll give you an overview of the challenges and a little bit more specifics. And from there, I will go to share some ideas that I have that I think may help in qualification and validation of the flow assays. So first, a little bit about the background for the ones who doesn't know that much about this area. So the T-cell therapies are generated either from autologous or allogenic starting material, which is it coming either from the patient itself or from other people who have donated starting material. And the starting material can be from different sources, for example, peripheral blood or tumor. There are several different types of T-cell products. We have the CAR-T, for example, Kymriah and Yescarta, that were, Kymriah was the first one being approved, and thereafter shortly came Yescarta. Today, there are several commercially available products for leukemia, lymphoma, and multiple myeloma. There are also a product called TIL, tumor infiltrating lymphocytes. We have TCR T cells, which are T-cell specific, antigen specific T cells, and we also have T regulatory T cells. Note that this is not an exhaustive list, and it's expanding quite frequently because the area is so quickly evolving. There are several clinical trials for these T-cell therapies in solid tumors. Recently also in autoimmune disease. For example, it was a very successful early phase CAR-T in lupus. And there are also, for example, in infectious diseases, for example, HIV disease. As I said, there may be more. I haven't, this is not a complete list. And what I want to highlight is that it's very important to know your product and to know your process. If you do this, you have quite high chances to have successful submissions. So flow cytometry is a method that is very important in the T-cell therapy space. It's a method that analyzes one cell at a time, and it uses lasers and fluorescently labeled reagents. And most commonly, it's antibodies that are labeled with a fluorophore, and the antibodies targeting different proteins on the cells. So if we have a very simplified workflow, so the sample is labeled with a fluorescent reagent, and once that is happening, the instrument needs to be set up according to all the procedures. And once that is ready, the cells are run through the instrument. It's coming through the flow chamber, and it's passing one by one the laser. And when the laser is hitting the cells, the fluorescent reagents are giving out the fluorescent light, which is collected into the different detectors that you can see here. And once all the cells are run and the instrument has collected all the information, the data is collected in an FCS file. It's a specific flow format for the flow data. This FCS file is then needed to be analyzed in a specialized software. And once you do that, it provides a percentage of the population, what you do, and the mean fluorescent intensity. So if we look at the dot plots down here, the histograms, so the first one here, it contains all the cells, and we point out, we select what parameters we want to look at, and we put the gate around the cell population that we want to look at further. We take that population and look at it in the next window. Here we looked at forward scatter, side scatter, and here we identify the live cells. And we take a closer look at these against, here we have the CD3 for T cells and CD16 and 56. And by doing this, we can identify our different cell populations. It's important to point out that one dot in these dot plots are equals to one cell. So what are the opportunities that we are having? In cell therapy, it's important to know your product, as I said, and we use it to determine the different T-cell phenotypes that can be in the sample. And different combinations of the antibodies to different proteins on the cell surface, for example, or inside intracellularly in the T cell, identifies the different phenotypes and what the type of T cells we have in there. For example, we can identify CD4 positive T helper cells, CD8 positive cytotoxic cells, regulatory cells, gamma delta T cells. And in addition to that, we can also determine their maturation state. The maturation state is what age there are, for example. So we look at them from stem cell-like cells to terminal effector cells. If we would compare this to humans, it would be from infant up to old retired person. We can also identify if these specific T cells, if they are exhausted or if they are activated, for examples. And depending on what instrument you have, because there are many different types of instruments and the number of laser the instrument contains, you can combine a high number of antibodies and markers per run that you do in the flow cytometer. In addition to looking at what type of T cells that we have there, we can also look at their function, what they are doing. Often in order to trigger the cell to react, we culture them together with a target cell type. So for example, if we would have a CD19 positive CD19 CAR T cells, which is a T cell recognizing CD19, we would mix that together with a target cell expressing CD19. which would then trigger an activation of the CAR T cell. And by doing this, we can investigate and characterize different cellular functions. For example, do the CAR T proliferate, or can it kill the target cell? We can look into cytokine expression and release, what's in there. And we can also look into signaling pathway activation, for example. And there are also other assays that we can do. We can look into up or down regulation of the cell surface markers, and we can look into state of apoptosis of the T cells, for example. All of this information we are using in the manufacturing of T cell therapy. In this example, I'm showing the common manufacturing process pathway of a CAR T product. So we start with the patient up here in the circle. So this would be an autologous product coming from the patient. That provides the starting material, the blood. We call that apheresis. After that, we select and transduce the cells, the T cells in there to express the CAR. They be expanded in the incubators, and finally we get the final product, which is then given back to the patient. So I am now going to show you where in the process where we use flow cytometry and what could be tested. This is very generic and not an absolute list. So the first thing is it's very common to look into the starting material and to characterize what we have in there. What type of cell types do we have? How many T cells? What type of T cells do we have in there? And how do they look like? We can look into, once we start the process, after the selection, we would like to look into how successful the selection was, and look into the percentage of T cells after selection. We would look into the expansion phase, and we can take a sample just to follow the expression of the percentage CAR, the chimeric antigen receptor, the expression, and also follow the percentage of the T cells if it increases or decreases, for example. For the final product, this is probably where you find the most testing, I would say. First, you need to do a release testing in order to release the product back to the patient. And it's commonly, flow cytometry is commonly used to look into the percentage CAR, percentage T cells, and you may have, for example, a potency assay that would be detected by flow cytometry, look into cytotoxicity and upregulation of CD107A, for just as an example. In addition to do the standardized testing for release of product, where you need to fulfill the criteria, you also need to characterize your T cell product. It's not for release of product, but it's additional information that you need in order to build the book of knowledge for your product. And here, you're very common to look into the different T cell phenotypes. You do some functional assays and etc. as much as you can to collect as much information as possible. Finally, the product is given to patient, and here you would look into the persistent, for example, the persistence of the CAR T cells in the blood samples that are taken from the patient. So you can see that flow cytometry is used all around the manufacturing cycle of the product for the patient. And therefore, it's a very important methodology that we need, and it provides a lot of opportunities to learn a lot about the product that you have and also the process. So we have all of those opportunities. What are the challenges? This is just I would say, limited list on the challenges, many common challenges. And of course, it differs from product to product depending on what you do. But the first one is you need to know your method and your assay. And you need to know, is the result really a true result? And in order to know this, you need to look into specific parameters and assess your assay. And depending on the phase of the trial that you are having and the purpose of the assay, whether it's a release assay or if it's a characterization assay, there are different requirements on what you need to do. So for characterization, you might need to have it scientifically sound or fit for purpose, that it's also called. And in early phase, before the pivotal trials, you need to have the assays qualified. And there are guidelines that instruct on what you need to do in regards to qualification. And for your GMP, for your release assay, it needs to be validated. And I will go further into this in a couple of slides. You need to include the proper controls in your testing. You need to know what is positive and what is negative for a marker or a protein that you have. And this poses a challenge, which I will also get into later on. Your T-cell therapy product is not only one type of cells, it's very heterogenic in the sample. And if, let's say, you have 94% T cells and then you have a few NK cells in your product, that distribution is not going to be exactly the same in vial to vial when you freeze down your product. And this causes a little bit of variation and makes it more difficult to assess your assay. You can have, it's very well known that it's when you set the gates that I showed before when I talked about the basics for the flow cytometry, it depends on the analyst when you set the gate and your panel that you have set up. So it's always a bit of subjectivity in the gating, and you always need to adjust the gate somewhat from batch to batch according to each sample that you are testing. You need to have proper instrumental settings and control for the spectral overlap from the fluorescence. This is called compensation. And you also need to have controls, and you can also encounter unspecific binding of your antibodies and other reagents. You can have issues with sticky cells or dying cells. And you can also have cells that are fluorescent just by themselves. It's called autofluorescence. All of this poses a problem when it comes to assess your assay to validate, qualify, and look closer into your assay. The current guidelines that are followed, that we are following when we are looking in qualifying and validating our assays, the ICH guideline Q2R1, they are not fully fitting the qualification and validation of the cell therapies. Because they are not updated with the information. I know that a lot of authorities are working on updating them so they would fit more for the cell therapy. But for example, there is no available reference material. A reference material is, for example, you can buy a vial of a this percentage of T cell or a this type of protein that you can purchase. But there is no such available reference material that is exactly the same that we can use. And as I said, there is a large heterogeneity between autologous products. Like my T cells are not looking like my colleagues' T cells, for example. You have mixed cell populations in the test samples. And specifically for flow cytometry, I will go through them closer in the next slide. But the fact that we don't have any specific standards and reference material poses challenges in the accuracy determination. And the linearity is a challenge because we rarely have a cell population of 100%. And many times there is an unspecific background, which also may differ between different batches. So it's a challenge to determine the limit of detection, the LOD, and the limit of quantification, which is LOQ. And we have also that it's extensive work that needs to be done because each marker and each cell population in the panel needs to be evaluated. This table on this slide, it's a lot of text. I'm not going to read all the text. I'm going to mention the most important things. But these are the parameters that needs to be determined in the qualification and validation. The accuracy, that is, if you have 100% viable cells, is that what you also measure? And here is the problem that we don't have a reference or a standard. The specificity is that we need to make sure that our antibodies is specific and the data that we are getting is specific for what we are measuring. And here we have the heterogenic samples that are posing a problem. And we also don't have standardized reagents all over. The linearity and the range of your assay, for example, the range is my assay is true for the data that comes out between 2 and 85%, for example. That's an example of the range. And the linearity, you need to be sure that you measure in the linear range of your assay. This is a problem because we do not have 100% pure population, so that might limit the upper range of our samples. If we only have 60% CAR positive T cells, we can only determine the range up to 60%. And the LOD and the LOQ, the LOD is the lowest amount of analyte that is detectable, but I can't put the number on it. The LOQ is the limit where I can start putting a number, a quantification of my assays. And here this is difficult because we have unspecific background that differs between samples. You might have autofluorescence, etc. And the precision is you need to make sure that it's repeatedly. If you measure the same sample at 5 different days or 5 different months from now, that you have the same results every time. So finally, I want to toss out some ideas that I am hoping may help you when you need to assess and work with your assays. The first I want to go through that there are some new tools available. New can also be maybe a few years, but I mention them anyway. For example, you can buy lyophilized PBMCs that are coming with a certificate of analysis where you have predetermined percentages of the different cell populations. This is very helpful for you when you need to, for example, have the same sample in each run to follow your assay. Nowadays, there are automated instrument that also comes with automated analysis. For example, to mention the Excelix instrument and the MaxQuant instrument. One really useful tool that I find is the synthetic cell lineage mimics from Slingshot Bio. These are synthetic cells, and because they are manufactured, they are always looking the same all the time. And I'm going to show you an example with this later on, how we can work with that. You can buy fluorescent particles that you can use to calibrate your instruments and also use as controls. They are called rainbow beads. And here's a very useful guideline for clinical flow cytometry that is published. Is the 862 guideline that is coming from the Clinical and Laboratory Standards Institute. So what I'm going to start with first is something very basic that is needed to be done and that is posing a challenge. And this I haven't mentioned previously in my talk. But every time you work with your antibodies, you need to decide what volume you need to know in order to have the most optimal panel and setting. For example, 5 microliter of your anti-CD3 or 2.5 microliter of CD4, etc., etc. And this titration is usually done using the cell types in your sample. But there is a problem because the expression of the marker may fluctuate or differ between your different batches or lots of cells. And I have going to show an example of that. And this is especially prominent if you use autologous or primary cells, for example. Cell lines maybe not so much, so that might be a good alternative. But it also may differ from cell bank to cell bank, working cell bank to working cell bank. And anytime you get a new lot of your antibody, a new titration is required in order to make sure that your panel is still optimal. So what you see here in the figures is just a vial of the true sites that we tested from Slingshot. And these are CD8 T-cell cell mimics that we used. And what you see in the next histogram here in the dot plot is just an example on how the antibody titration can look like. On the y-axis is the mean fluorescent intensity, the signal, the strength of the signal that we get from the fluorescent antibody. And then on the x-axis, we have the different volumes that we tested. And to the far left here is the sample in itself without any antibody added. The graph to the right is showing the compiled data that we got from this antibody titration. And what we are looking for is the volume where we have the best separation between positive and negative. This can be done in a few different ways. And here we are looking into the ratio between the positive and the negative signal of the mean fluorescent intensity. The blue dots is the negative signal, and you can see that it's very close to 0. The red squares here is the positive signal that we get, and it would be the ones up here. And the purple triangle curve is the MFI ratio. So what we did in this experiment was that we had our old lot of a CD8 antibody that we had titrated on peripheral blood mononuclear cells, PBMCs. This old antibody we titrated again on a different lot of PBMC to what we used first, and this is traditionally what we have done. You use different lots of cells to titrate your antibody. We also tested it on the true site to see how the expression looked like. And here we have the new lot of the antibody, which we tested with the new lot of PBMC and the true sites. And then we can compare how the expression looks like and the titration looks like if we do it on PBMCs or true sites. The hope is that we can use true sites to continue to titrate all the lots that comes afterwards, because the true sites, as I said, it's a cell mimic. They are always looking the same. They always have the same expression on the CD3 cells. So this would be an excellent tool. This is just showing how the titration of the old lot of antibody, the first time we did it on the PBMCs, and the histogram here, we have the CD8 MFI signal on the y-axis and the antibody concentration on the x. And here we have the graph with the results compiled. And you can see the top of the highest point of the MFI ratio positive to negative. And this was found to be at the volume of 0.625 microliter of antibody to the 100 microliter of sample that we had. We then took the same old lot of the antibody and we tested that on a different batch of the PBMCs later on after 6 months of use. The graph to the left show you the first titration where we had the 0.625 microliters. And the graph to the right show you how it looked like in the new PBMC. And what's within the green rectangle is the result obtained at 0.625 microliter. And you can see that this data point is not the highest of the curve. It's actually the next data point. So we can see that it would not be the optimal to use 0.625 in our, with our new lot of antibody. It's good, but it's not the best one. And you can see if you look at the MFI, and you have the signal here at 20,000, you can see the positive signal is going up to 20,000 MFI, and the MFI ratio is also only going up to 20,000 MFI, while for the new lot of the PBMCs, it goes up to over 40,000. So here you clearly see that the different batches of the PBMC differ in the expression of CD8. So on this slide here, I present the data that was done by comparing the titration if it was done on PBMCs or on true sites. So the dot plot here, the plot to the far left, shows the old antibody lot, the volume of 0.625, tested on the true sites, the cell mimics. The new CD8 antibody lot is here on the middle graph. And you can see we tested it at 3 different concentrations. And the one that is the lowest is the unstained sample. And when we plot that in our graph, so you can see that this is the MFI on the old antibody, while the green, you see the MFI at the 3 different ratio that we had. So you can see that the new antibody lot has a lower signal MFI compared to the old antibody lot. So therefore, we decided that it's not optimal any longer to use 0.625 microliter. So we did a calculation. The MFI was 87% of the MFI ratio, and therefore we adjusted the volume accordingly. So the new volume would be 0.72 microliter per 100 microliter of sample. And this we will continue with to use for the CD8 antibodies that we are having. So this was the first example, and now I'm going to go into the second and the last example that we have, where we looked into how we can use a product from Slingshot that are called HYPAR comp beads. And you see the bottles down here. It's a compensation bead that is used for compensation and setting of your assay when you run it. They come in 2 vials. One vial is 100% positive, and the other one is 100% negative. And as I said, we want to measure the linearity to make sure that the assay is linear from 0 to 100, or at least knowing what the linear range is. The LOQ is very important to know because it's the lowest reliable quantitative measurement in the method. And so what we did, that many times you have your cell type, for example here, the T cells, and in order, if you have a sample with 100% T cells, you need to dilute it down to 80, 60, 40, 20, and 0% when you assess the linearity. It might be difficult sometimes to find a cell of different cell type. Many times cell lines are used, but they have slightly different properties many times to what a lymphocyte is having, and this may pose a challenge and may not be the best option. So what we did here was that we did this in two ways. First, we had our product, a T cells tumor-infiltrated lymphocyte that was very close to 100%. We had this sample, and then we diluted the the 100% cells with the negative hypercomp beads that I call naked beads because they are not binding any antibodies. And we diluted it down from 80% down to 0. We did the same thing using the 100% hypercomp, the positive beads that are binding all your antibodies, and diluted them in the same manner with the naked hypercomp beads. And with this, we assessed the linearity and the LOQ. So I will walk you through this slide. The top two graphs is the linearity assessment. Here we look at all the concentrations that we diluted down to. We used all of these samples to look at the linearity. When we assessed the LOQ, we used the 20% down to 0 to assess the LOQ because the upper end is not of interest in this case. The lower graph is showing the results for the LOQ, and the two left is showing you the hypercomp data, and the two graphs, upper and lower to the right, is showing the T cell data. I think this provided a lot of good information. Looking at the hypercomp and the T cells diluted with the naked hypercomp beads, the linearity was excellent. The R-square value, which you often look at, is for the hypercomp 0.999. And for the T cells, you can see here, if you look at the data points, they are deviating somewhat, but the R-squared is 0.991, which is also very good. It's when we're coming to the LOQ that they differ somewhat. The hypercomp beads are very clean, and they are the same all the time. They are very constant in your replicates. And we did this with replicates, and we assessed the LOQ by looking at the percent CV, coefficient of variation, between our replicates. And if the percentage CV in this case here was higher than 10%, then we said that this is where then they were excluded and not included, because then we say that they are not reliable. So the LOQ for the hypercomp, we could set to a little bit more than 1%. When we look at the T cells, you can see that you have a lot more variation from your regression line here. And this is because it's cells that we are working with. You know, it's not completely the same distributed, and you always get some variation when you work with real cells. Here, the LOQ, if we would have put that to 10%, we would have ended almost at 20% TIL to be the LOQ. So here, because it's a feasibility, I don't have any predetermined criteria. I said, okay, a CV of 20% will do. So the LOQ was put to close to 3%. This was the first time we tested this, so it was a feasibility study, and you do feasibility studies to know your assay. So what I would do to repeat this, I would increase my replicates for a better assessment than what we had here. And I think that I would choose the option to use the cell mimics instead of the hypercomp beads. So when I have the T cells, I would probably buy a B cell mimic, for example, to dilute my T cells with. And I want to finish up with just summarizing the opportunities that we have for flow cytometry, because I don't think that we could do cell therapy without it. It provides information and knowledge of the product from the starting material to the final product to the patient. And we, in addition to get information about the product, we also get information about the process, how well controlled the process is. We can identify different cell types and the many phenotypes of the T cells that we have. And we can also assess many functions that the T cells are having by using flow cytometry. My last advice that I have learned over the years is that in order for successful submissions, you need to know your process, you need to know your methods, and you need to know your product. If you know this, you are very good off. By that, I want to thank Minyak Duong, who is an senior analytical scientist at Hygen. Excellent flow cytometrist. I don't know what I would do without him. And he's the one that ran the flow for the ideas that I presented to you. And of course, I want to thank Slingshot Bio and LabRoots for organizing this webinar. And thanks to all of you who has listened in.
A special thanks to you as well, Therese, for a very informative webinar. And again, to the Tygen team, Mignon included. It's just really showing your expertise and the expertise that you're bringing to this field. So with that, I'd like to introduce some of our panelists. This is a point in time when we're moving to the Q&A. So those of you who've been attending the webinar, if you want to go ahead and put any questions you may have for this group of panelists into that question bar, Q&A bar, we will be monitoring it and bringing those in alongside the pre-selected questions that we have ready for today. So it is my pleasure to introduce a few more individuals to you. On our panel, we have Hiba with us, Deng Heidi. Hiba comes to us with a rich background, both serving on the FDA, CBER, and OTAT. In addition to Hiba, we have Virginia Litwin. Virginia Litwin is very well known in the clinical flow cytometry and I would say CRO flow cytometry space for her work, including most recently her contributions to the CLSI HI62 guidelines. One of her partners in crime, in addition to Hiba, is Lily Wang. Very honored to have her with us. She serves NIST and actually has been played a very pivotal role overall in the standardization of flow cytometry. So assisting in the writing of some of those guidelines, other guidelines, in addition to the HI62 guideline. And then also really my honor to bring Daniel Baker onto the panel. He has a recent contribution, many contributions, but a recent one in Nature where he really outlines the future of cell therapy as the therapeutic for us moving forward, where the cell is the drug. Brings a lot of passion, comes from Carl June's lab at Penn. And so really wanted to bring this diverse set of panelists to you, the audience, because I think in my limited experience in this space, I think if we're going to move forward and be able to bring in some of the regulatory guidelines that may be coming in the future, that we should do that with all voices included, and that we should do it in a very strategic way. And so I will actually start with the question from the audience first. The question was sent in by Wei Ann, so thanks for this question. For a flow cytometry assays used in CAR T products, specifically in manufacturing, what type of regulatory requirements do they need to meet currently, and what regulatory guidance do they follow? And And I'll kick that first, I think, to you, Therese, with letting the other panelists jump in as well. So are you aware of any guidelines
Absolutely.
regulatory guidelines? Thank you.
The one of the major big guidelines that we are following when we design our validations and for CAR-T is the ICH Q2 R1 guideline. That is, I would say, the most important for us that we need to follow.
Okay, thank you. All right, and the next question comes from Phil Robertson. Thanks for the question, Phil. What are the implications of not using precise and accurate flow controls within cell and gene therapy? I don't know if you want to start with that one, Daniel. We'll start on the research side, right? If you don't have those precise controls, and then we'll work our way to you, Therese, and then we'll have, I don't know, Virginia, Lily, or Heba step in.
Yeah, I can take it. So, Phil, I think it's a really great question. You know, cell therapies are an entirely distinct platform of therapeutics. And one of the reasons why it's so important to have these very stringent and precise controls is it's not the same thing as having, 1% of a drug circulating in your system. If you have 1% of your blood cells or T cells being CAR T cells and you don't know that, that's a dramatically, whether it comes to toxicity or efficacy, that matters a lot. Especially as we're starting to think about, decade-long studies, we're going to see very small proportions of these subsets potentially being CAR T cells. And as we're trying to track these over potentially a patient's life, it's super important for us to be able to understand kind of exactly what's the magnitude of the effects that we're searching for. So I think, especially as these therapies are getting more refined and as they are broadening, it's really important for us to be able to ask these questions in a very careful and precise way.
And Therese?
Yeah. Thanks, Daniel. That's a very good summary. I think it's very important to be sure that what you measure is the true measurement, that it's the right measurement. If I would measure my sample at the next day or 5 days later, am I going to get the same result? Because as Daniel said, it's very important to know what happens in your body. And if we look into the release testing of the product, we need to be sure that what we release to patient is safe and that it's consistent and meets all the criteria that are set up for release of that product.
Yeah, great. And with that, Lily, I'm actually going to come over to you because I know controls has been something you've been very passionate about for your full career.
Yes. Can you hear me?
Yes.
Yeah. Okay,
yes.
Great. So actually, there's a huge push, for standard development. That including both the material, biological reference material, and also measurement procedure. I think that pave the way to documentary standard more for fit for purpose. For example, CAR T therapy and engineered cell therapy. So I think with that said, I think the industry, especially manufacturer, really should know get in touch with a standard organization. For U.S. part, certainly needs to drive standard development, both reference material and also documentary standard, along with CLSI. Certainly, Virginia is on the in the panel list. So NIST really have flow cytometry standard consortium that's really address the lack of reference material and a fit for purpose reference procedure, really driving the instrument standardization and assay standardization. So I guess what my recommendation would be, we work towards that end as the process progressing certainly we want more informed the industry or basically community, not only including our consortium's product, but also we work with the international organization, for example, UK's NIDSC. So all those related work really addressing the lack of standard.
Yeah, thank you for that answer. And Virginia, as I send it over to you, I'm actually just going to bring up the point that cell therapy has certainly elevated the need for flow cytometry in the analytical development, late stage manufacturing process of drug development. But as you've alluded to previously, flow cytometry has been part of drug development for decades, well over 2 decades, and has been really actually integral in setting up any biologic drug and other therapies. 90% of all drug development starts with flow cytometry. So would love to hear your answer to this question, not just as it pertains to cell therapy, but as it pertains to drug development overall.
Great, thanks. And thank you, Therese, for a really good presentation. You touched upon so many critical things. So I don't know where to begin, but just circling back to what Daniel was saying. And in CLSI H62, we address the basic approach that you would use for flow cytometry. And Therese picked up on the points, and she's sort of new to this document. So I'm really pleased to hear that it sort of validates what we wrote in there. And what Daniel's talking about, validation has its own parlance, but is we need a specific assay so that we know we're tracking those cells. And we need a sensitive assay. And so we know that we're picking them up at the level that we need to be seeing them. Like, is it okay to see 1%? Do we need to go lower than that? That type of thing. And the other thing we talked about in the document, and that Lily just touched upon, and Daniel as well, we're going to monitor these patients for a long time, and therefore we need to have control of our assay. And in that regard, it's not really specific to flow cytometry. We just need good old-fashioned quality procedures. But what is specific for flow cytometry is, how do we monitor that assay? How do we get a control material that will allow us to know how the assay is performing over time? And it's a bit tricky with flow cytometry, because if we have cellular controls, they're going to have a limited lifespan. That's why I'm a big fan of the Slingshot product, that we can have a long-lived control material. So we're not constantly changing lots of control material. And that really helps us assess the assay longitudinally. And also, as Therese was talking about, helps us verify that, yeah, we got a new lot of antibody and we're still getting the same result with our QC. So we think our assay is still stable. So there's a ton around this. And I think one thing, building also on what Lily said and what we did with H62, is it was a mix of biomarker people and clinical laboratory people, and that there aren't guidances. There weren't, and that's the best we have to date. And so what we came up with, I was representing AAPS, and there were clinical cytometry people, is we need to write them with the people that know the technology. So as we progress with cell therapy and we're using flow cytometry in manufacturing, then yeah, we need Therese to come join us and along with NIST and the other groups, and see what is it we have and what is it we need for this new application. We always talk about context of use, fit for purpose. Well, we have a new purpose, so we need to really look at that and come together as a community to do that. Sorry, long answer.
No, it's a great answer, though, Virginia. Thank you for that.
Can I?
Hiba, I'm not sure. Go ahead.
No, sorry. If you want to take Hiba first, I can jump in later. Yeah.
Yeah,
I'm
sure.
just going to check
Sorry,
in
I
with
have
Hiba.
problem with. Can you hear me?
Go ahead.
Yeah,
Yes.
sorry, I have problem with my camera. I don't know why. Everything set up, but yeah, to answer the question regarding the controls, so really controls are critical, and including like controls for instruments, controls for your assays, also reference reagents, and also a reference standard for quantitative flow cytometry. So these controls are critical because it show you how you accurately identify the critical quality attributes for your particular product, and also how well you can characterize it and also identify the release parameters. Also, it will show you like how potent is your product, and you can correlate it with clinical efficacy. So controls in general are very essential, and it's a wide term. You can use it for every single variable in your flow assay. So we have a collaborative, as Lily mentioned, we have, a collaborative studies with under the Flow Cytometry Consortium to develop biological reference reagents and reference standard for many assays. We also collaborate with many other institutes for reference reagent for different cell types. So in general, controls are very essential and critical, and it should be introduced in, as early as possible when developing your assay, so you have a tight control on your variables as well.
Thank you, Hiba. Very nice answer and very accurate. And with that, I'm actually going to kick it back to Hiba because just controls are necessary. Leanne is asking us, she says, nice presentation. Using the slingshot beads for linearity is a very interesting approach. I want to make sure that I understand what was done. You use the slingshot product with the antibodies alone to see if the reagents are performing with linearity. So more of a device control for the assay and not the linearity of the assay in context of a product. If you, Therese, don't mind addressing that question, and I can repeat it if it's needed. And to the audience, we will be running just about 5 or 10 minutes over. Just I know time check, we're getting close. So go ahead, Therese.
No, that is a really sharp observation, and that's why we tested it both on the hyper beads as well as we tested it to mix it up with our cells, just to see what the difference was. I think it's important to have control of your instrument and the linearity. You can do that with fluorescent particles. But in regards to the assay, as to say, I think that you need to use your cells of interest that you are looking at, and then you can use the slingshot cell mimics to dilute them, because you need to dilute them out, so to say. Today, I know you need to be very, have a good imagination when you design your experiments. What are you going to dilute your cells with so you get your percentage down of your specific cell populations? And because everything you have in your assay, antibodies, reagent, matrix, flow buffer, anything might have an impact. So that's why I think you're absolutely accurate that it's better to use the cell type you have, and you can dilute it with, for example, the slingshot cell mimics that I like, because they look like lymphocytes, they behave like lymphocytes in the flow cytometer. So I would prefer to use that instead of a cell line, for example.
Yep. And with that, I'll answer one other question. How is our lot-to-lot consistency? Our lot-to-lot consistency is superior. We have some collateral where we can put it up and show you, but we're for most products under 5%, it does matter what the surface protein is, but we certainly have that lot-to-lot consistency that's ready for you to see because of some work that we did with NIST on a product that's called TBNK. So that's a good question. Another question, and I will come back, is will following the CLSI H62 on its own meet the ICH-Q2 guideline? And this is from Rebecca.
So what we say, and I don't know the ICH, I don't know the ICH guidelines. So the H62 is addressing how to develop and validate a flow method. And it also talks about, which we I don't know if we touched upon too much today, but how we standardize our instrument, which of course is critical. So this would be for your assay. And then the ICH guidelines, I don't know, Therese can comment on those, but so it's specific around the flow cytometry data, but it's agnostic to the method. So we wanted a guidance document at the first pass to say, okay, you decide your context of use. You will need to talk to the regulators if required and say, this is our approach. And going back to that last question where Therese said, you need to be creative. You do. You know, we don't have standards that we can spike in and use. And so you would write your validation plan, look at your approach, describe it, talk to the regulatory agencies and if that's applicable, and see if they agree with that. And so I got one question early on saying, if I follow this, will the FDA approve my assay? And it's like, you know, you need to talk to them because they may have other requirements, but at least you have a well thought out approach.
Yeah, I do think, Rebecca, your question is very good. And I think, and I'm going to let anyone else on the panel who wants to speak to it, but I think it does bring up some homework maybe for this group is putting those two guidelines next to each other and seeing if they could stand alone or if they actually need each other. I don't know if anyone else wants to comment.
I want to mention, I can comment on that because I think the 862 guideline is, like Virginia saying, it's more, you know, recommendations how you do it specifically for flow cytometry. The ICH Q2R1 guideline is not specific for the flow cytometry, it's specific for your assay validation. It instructs, for example, it explains what accuracy is and what accuracy, slightly how you should do it, but it doesn't have any specific instructions like the 862 more practical instructions. The ICH Q2 guidelines are giving you, this is what it is, and this is how it should, what it should measure, and this is the outcome that you should report. But it doesn't tell you how to do it. So I don't think that you can take one or the other. You need to fulfill the ICH Q2 guidelines according to what each parameter in your validation should deliver. And I think that the 862 is complementary because it kind of gives you guidance on how to deliver it. Virginia, I think that would be, or Lily,
So if I can jump in then. So
that's how I got to use it because I found it really instructive.
So that's exactly why what it does, Therese. So is it says, so the other's telling you have to do accuracy, and then we go into quite a bit of detail on how can you do that in flow cytometry, looking at, you know, in what we do. And the same thing, other things where limits of detection capabilities, limits of quantitation are addressed differently. So this is how you address it, but maybe the ICH guideline is telling you what you need to address. And the same thing was for the CLIA guidelines. They were equally instructive, just saying do accuracy, do precision. It's like, well, how? You know, and there's a lot of challenges with flow cytometry.
Exactly.
I agree with you know your both assessment, nice assessment. I think H62 really address more specific how you really develop an assay and validate an assay in really great detail, including instrument longitudinal monitoring, and also assess your instrument, how what's the sensitivity of the instrument, how you want approach cross instrument more standardization. So those are all touched upon on the CLSI H62. So that's part of necessarily work closely with the CLSI in terms of documentary development.
All right. Well, this has been a really great panel. We do have every intention of moving forward and having more discussions like this, just because you can tell we don't even have enough time to cover. There's many more questions I haven't had a chance to get to. The final question, though, I'm going to kick to you, Daniel. Just speak to us a bit about where the future of cell therapy is going and what maybe the next 5 to 10 years looks like in the clinic.
Yeah, I think it's a really exciting time. You know, if you went back a decade, it would be really hard to predict what has happened. And it's really clear that there's a huge amount of inertia and enthusiasm, even, you know, all of you being on a call like this. And so it's clear that cell therapies have been transformative for blood cancers. And maybe just to touch one of the questions that we didn't get a chance to touch, there's a whole bunch of intricacies here that will be teased out in the next couple of years. Like on a basic science side, we're seeing the percent CAR positive is super important. Engineering these cells with to be smart CARs or armored CARs, this is going to change the way that the field is asking some of these questions. As we go into different diseases, early stage autoimmune disease, and then throughout other chronic diseases, I think it's going to be really important because these are truly living drugs. They're going to respond, they're going to react, they're going to be different. And so as we understand these questions about T cell exhaustion and CAR T cell dysfunction, how do we make stronger and better armored CARs? All of these questions are super fundamental. And so being able to actually characterize these cells both as they're going in and as they're responding and persisting in patients is super critical. So I think it's an exciting time for the field, but I think there's a lot of work still to do to understand what's actually happening in patients and how is this going to be impactful for medicine at large.
Thank you. And thanks everyone for your time today. The remaining questions that are there that are unanswered, we will do our best to get them answered and sent out to you. We really appreciate everyone's time and look forward to version 2. Thank you very much.
Thank you, everyone.
Thank you.
Thanks.